So what is iGEM? iGEM stands for International Genetically Engineered Machine. It is a competition based on the open source for undergraduate and graduate students in the field of synthetic biology. Each year, multidisciplinary teams composed by students and advised by researchers undertake during 6 months a project from scratch and have to take into account the economical and ethical aspects of this project. And that's why having people outside of science is a valuable asset. So the competition ends in Boston during a big conference where students present their results. And this year, 2015, 280 teams attended to this conference. So what does an iGEM do during 6 months? We started our project in January when Florian Cherrier from the Pasteur, for the Institute, proposed the idea to have their own team, and shortly after the team was composed. We work on all aspects of our project because iGEM is more than building a biological system. It is also about communicating, finding collaboration with other teams from the iGEM competition. It is finding sponsors, that's quite difficult. And it is also making the public aware about the problem you wanted to deal with. But the main objective is to address an important issue and to generate the tool to deal with it. And the practical aspect of that is building BioBricks, which is a design to make these tools accessible to the scientific community and to the industry in a standard fashion way. So, what is a BioBrick? I have here a nice laser. A BioBrick is here the whole part. It's composed of a plasmic backbone and here an inserted sequence called BioBrick part. This sequence here is the only variable section of the whole BioBrick. It is framed by 4 restriction sites which make the thing that the plasmid needs to conform to a restriction enzyme assembly standard. And that means that was quite difficult to get, but that these restriction sites have to be absent of the BioBrick part. And the whole plasmid is the only way to standardize a plasmid for the iGEM competition. This final BioBrick is then registered in the register of standardized biological parts, which is open access. That means that each BioBrick is really easy to reuse because it's first standardized and then open access. So actually this BioBrick then contains the genetic information about the project of each team. And for the iGEM Pasteur team, this year, we focused on an important issue, which is your holiday. Yeah. That's how you expect your holiday to be next summer. Paradise on Earth, crystal clear water. But that's far from reality. This location actually looks like this. And that's the reality of our oceans as I speak. Pollution by plastic particles is one of the major issues of this century. And as such, is a global problem. This map represents how critical the situation is. Here on the screen, each white dot represents 20 kilograms of plastic. So I can distinguish one single dot, and I'm closer to the screen than you, so I think it's quite difficult for you. 90% of all these particles are smaller than 5 millimeters. That makes it impossible to clean and small enough to get into our food chain. So how do we deal with that problem? Now, here's something interesting to know. 75% of all this pollution comes from onshore activities. So here's where we can start. I took the example of Europe. Europe gathered some of the wealthiest and most developed countries on Earth. So it should have the money and infrastructure to deal with in a clean way with plastic waste. Each year it deals with 25 million tons of plastic waste. It recycles a bit more than a quarter of it. 36% are burned to produce energy, and almost 40% are burned into the ground. That's a nice solution, I find. So here's where the iGEM team step in. We thought we cannot go on like this. So we tried to find a solution to treat our parts of the plastic waste. And we came up with a concept we named PlastiCure. Here's the principle of PlastiCure. PlastiCure is a biological system able to degrade plastic waste, but nothing new so far. It is also designed to reuse the plastic degradation product to then synthesize a biologically active compounds. To do so, we want to use E. coli as a heterologous host and engineer in E. coli the degradation pathway and the biosynthetic pathway. Now, the big challenge of this project is to make these two pathways work simultaneously. And that's all about the project during the summer. The whole iGEM team has 3 months to find a solution to this. The idea here was first to create a system able to find a new answer how to treat our plastic waste and to produce an interesting compounds with a high value that will increase the efforts of plastic recycling. So, such a big project needs dedication of many people, and therefore I would like to thank our advisors, which are really helpful. They are there every day to help us, to guide us, because we're just undergraduates, but also graded students. I want to thank the Pasteur Institute, because they granted us access to their facilities, and for their undeniable support, along with our sponsors, Institut Pasteur, Genius, Zone 30, who is here, and also to all my colleagues who make this project possible. Thank you very much for your attention.
On est très fiers de discuter et communiquer avec des vrais chercheurs dans le monde de la science.
Première grande conférence, déjà, peu de stress, mais arrivé sur scène, on oublie tout en fait, c'est assez impressionnant.
Et on nous a invités même pour présenter les résultats, donc on espère qu'on pourra un jour présenter les résultats positifs.
Ouais, c'est notre objectif.
Merci, du coup, on continue à travailler la partie scientifique à fond, on continue à chercher des sponsors et on continue à s'amuser.